Uh, Welcome back. In this session, we will be discussing the chemistry and medicinal aspect of neurotransmitters. Okay. Now, neurotransmitters are chemicals, small chemicals that are generated from the neurons, and then they bind, they have a receptor site very close by in the next adjacent neuron or muscle cell, and then bind there and then send a signal. Okay. Signal, and then that creates a cascade of reactions. So, they are called neurotransmitters. So, neurotransmitters are small molecules. They are also belong to a class of what are called chemical messengers. Chemical neurotransmitters are chemical messengers which are secreted by neuronal cells, and then they have a nearby binding pocket in adjacent neuronal cells or muscular cells, okay, and then produce some signal which then carries out a cascade of reaction and exerts the effect, the desired effect. Okay. Now, chemical messengers are basically two types. One is neurotransmitter, another is hormone. So, neurotransmitter already I gave you the definition. The hormones are usually secreted from the glands, and then like pituitary gland or adrenal glands, and they actually traverse through the bloodstream to different sites, and then exert the effect by binding to their receptor. Okay. So, the difference between chemical messenger and hormonal is there where are they produced, number one, and number two is that neurotransmitters are basically they do not traverse long distance, they bind to a nearby site, whereas hormones are distributed to different parts of the body. So, today we are going to talk about a neurotransmitter which is called acetylcholine, okay, which is called acetylcholine. Now, the structure of acetylcholine is given here, very simple. It is an ester, and then it is basically an ethanol amine, but the amine is present as a quaternary salt. There are 3 methyl groups here, okay, 3 methyl groups. So, it is a ethanol amine derivative, and the alcohol is esterified by the acetyl group. So, that is called, hence the name acetylcholine. So, you can say what is choline? Choline will be OH and then N, this 3 methyls and plus, this is what is only choline. Okay. Now, this what is the function of this acetylcholine? Okay. That is number one. So, the questions that are that what is the function? Number two is that where does it bind, means where does it bind? It has got definitely a receptor and a binding pocket, but exactly what is the characteristics of that binding pocket? Number three is how is the concentration of acetylcholine maintained. Okay. So, these are the few questions, and then what are the medical consequences? Because now we are going to the topic where we will pick out different molecules, and then their chemistry or biochemistry, and then accordingly we will try to design drugs. The molecules that we will be talking about, either they are shortfall or they are excessive secretion, will cause some harmful effects. So, you need medicine to treat that. So, these are basically, first of all we have to see the chemistry of this acetylcholine, and accordingly we will then decide what are the molecules that can modulate the function of this neurotransmitter. Okay. So, what are the medical consequences? That is the fourth question. Now, acetylcholine, as I said, all neurotransmitters are secreted by the neurons. Okay. Now, acetylcholine has receptors which should be nearby, that means a nearby neuron, or it could be a muscular cell. Okay. Now, suppose if it is this is suppose your neuron. Okay. So, acetylcholine is secreted from here, and that secretion is dictated by what is called an action potential. Okay. When to fire or when to generate acetylcholine, that depends on this action potential, and then you have adjacent neuron suppose, and it has got a binding pocket here. So, acetylcholine, this is called the postsynaptic cleft, postsynaptic cleft. Okay. So, acetylcholine drops into this area, and then it goes and binds to this active site pocket, and that creates a signal. So, what type of signal, that also will highlight a little bit. What has been found that acetylcholine has two types of receptor sites. Now, these receptor sites are named according to the neurotransmitter or according to the hormone, like estrogen is a hormone. So, where it binds, it will be called an estrogen receptor. Similarly, the receptors where acetylcholine binds is called cholinergic receptors, Now, these cholinergic receptors again are two types. In one type, what happens that there are some natural compounds, like this is what is called muscarine. This natural product muscarine, if it is taken, then this muscarine goes and binds to a particular type of receptors which are meant for acetylcholine. Okay. So, they are called muscarine receptors. They are called muscarine receptors, but they actually belong to the cholinergic receptors. So, cholinergic receptors are those where acetylcholine binds, but they are subdivided into two. One is called muscarine receptors, that means one is distinguished by the fact that only those receptor sites can be occupied by muscarine, and the other is the site which is called nicotinic receptors. Nicotinic receptors, and nicotine is shown here. This is nicotine and that is muscarine. So, nicotine will not bind to the muscarine receptors, or muscarine will not bind to the nicotine receptors, nicotinic receptors, okay. But both belong to the cholinergic receptor systems. Now, what is the signal that we are talking about? What it does actually when acetylcholine binds? What it does? There are two kinds of receptor already we know. Now, in one kind it is the nicotinic receptor. What happens that in the cell membrane, you know, the cell membrane is basically a lipid bilayer where the hydrophobic part are pointing towards each other, and that actually constitutes the membrane. That means, the inner part of the membrane is very hydrophobic, and then these are the head groups. So, one is pointing towards the outside, and another is pointing towards the towards the cytosol. Suppose this is the cell, so these will be pointed towards cytosol and these head groups are pointed outwards. Now, in these membranes, what has been found that membranes, there are many proteins which are embedded in the membrane, and these proteins are called membrane bound proteins, and these membrane bound proteins have an exterior surface which is interacting with, see, this is your cell which is interacting with external molecules, and it has also some internal part, interior part which is pointing towards the cytosol. Okay. So, now the question is, what is the purpose of these different membrane proteins? They have different functions, but one type of function of the membrane proteins is that they allow foreign molecules to enter through them by forming channels. Okay. By forming channels, that means, if this is your, this is the, suppose the membrane, and suppose this is the membrane protein. Okay. Now, what it does, that it can allow, it can provide a channel or a pore through which foreign molecules or ions can enter into the cell or can go out of the cell. Okay. Now, and these pores are, sometimes they are closed, sometimes they are open. When it is required that some ions need to enter or get out of the cell, then it opens up, and that opening and closing. So, basically this is acting as a lock gate, and the opening and the closing can be dictated by several ways. One is called mechanically gated. Mechanical means you apply some pressure from outside and that will open the gate, like this membrane protein. This is the channel. So, the channel is open when you apply pressure. If you release the pressure, then the top portion join with each other and close that pore. Okay. So, this is called mechanically gated. That means, the gate is open only when there is a pressure from outside, mechanical pressure. Then there are different types. Voltage gated, that means the electrical potential controls the opening and the closing process, but the one that we are interested in here is what is called ligand gated ion channel. Okay. These nicotinic receptors are basically ligand, they activate the ligand or they act as the ligand gated ion channel opening. How? Because this, maybe the next slide will have that. Let us no, this is the different one. No, I think this is, I can explain it. Here it is written, nicotinic acetylcholine receptors are ligand gated ion channels permeable to sodium, potassium and calcium ions. So, that means they allow the inflow or outflow of these ions. Okay. In other words, they are ion channels embedded in cell membranes capable of switching from a closed to an open state when acetylcholine binds to them. In the open state, they allow ions to pass through. So, what we are saying that, suppose this is the that membrane protein. Okay. Membrane protein, there may be different, it is a multimeric protein, it is a multimeric protein. So, inside there is a channel. Now, the channel is something like this. So, that means this is basically very narrow, means this is closed, suppose. Now, it has this membrane protein has a binding site, has a receptor site where acetylcholine binds. So, when acetylcholine binds, this channel becomes, the membrane protein will be like this, sorry, like this. So, now it becomes, sorry. So, earlier it was going inwards and locking the passage, blocking the gate. Now it becomes open, and now the ions can freely move into the cell or outside the cell. But this will happen as long as acetylcholine is bound to the receptor site. So, as soon as the acetylcholine is detached from here, this goes into the resting state, that means the closed state. Okay. So, this opening is by, I can say, ACHE, that is what acetylcholine you can abbreviate as ACHE, acetylcholine, acetylcholine, not E, sorry, only acetylcholine. So, what we have learned, that there is this, when you talk about the nicotinic receptors, here the nicotinic receptors are what? They are ion channel, they are ligand gated ion channels. Okay. Ligand gated, ligand means here ligand is acetylcholine. So, it sits on the membrane protein, which are ion channels, ion channel proteins, and then it opens up the channel and the ions pass through it. What ions? Sodium, potassium or calcium, and then when it is detached, then it goes to again the state where the gate is locked. Okay. So, basically it controls the passage of sodium, potassium or calcium. Now, the question is, who controls, what really controls this? See, basically now you have, remember, there must be a neuron here which drops in the acetylcholine into the synaptic cleft, and then that goes here. So, there is an equilibrium between ACH plus this receptor. So, that will have an equilibrium process, ACH receptor, that is the bound state. So, in the bound state you have the open gate, okay, open gate, and here you have the receptor closed here. Okay. So, there is this equilibrium going on. Now, if you want to see, you do not want the signal to be produced for a long time, because that is also not good. It has to be maintained properly, that when it is required, acetylcholine drops from the neuronal cell in the synaptic cleft and then binds to the to its receptor and opening the gate. But this because there is an equilibrium, but you want this signal to be produced for a certain length of time, and then it should be stopped. You do not want all the time. So, what will happen? How to stop that? You have an enzyme called acetylcholine esterase. That is what is ACHE, acetylcholine esterase. This acetylcholine esterase, what it does? That, remember, acetylcholine has a structure O, CO, CH3 and then N, Me3 plus. So, acetylcholine brings it back to the choline, Okay. So, again just repeat the whole thing. Acetylcholine sits onto the nicotinic, if it is nicotinic receptor, then it sits onto the nicotine receptor and then opens up the gate. This is called ligand gated ion channel, and then the ions move in or out. Okay. And then how to stop this signal? Because if then you have to either hydrolyze, if you hydrolyze this the acetylcholine by this enzyme acetylcholine esterase, then the equilibrium will be shifted on this side. Okay. So, you will have you will have the resting state of the receptor, that means the closed, where the gate is closed, and that means the flow of ions will also stop. Okay. So, this is for the nicotine receptor. We will come back to this acetylcholine esterase once we are through with the muscarinic receptor. The muscarinic receptor is little bit different. Here, this is the membrane protein, and in the membrane protein, remember, the receptor site is in the membrane protein. So, also that channel, ion channel, the channel is also maintained by the membrane protein. There is another type of receptor which is the muscarinic receptor. What it does? It has got these different monomers of the protein. So, it is a complex, it is a multimeric system, and then what happens when the ligand, in this case again acetylcholine, if it is released and sits onto its binding pocket, this is, remember, this is your muscarinic binding pocket, muscarinic receptor. Okay. So, when it binds, what happens here? This is what is called GPCR, G protein coupled receptor, G protein coupled receptor. This is a different kind of receptor. It activates the G proteins. Now, what is, so earlier when the ligand, means acetylcholine, sits onto the nicotinic receptor site, then it is a ligand gated ion channel. In the muscarinic site, it sits onto the membrane protein, which is a G protein coupled receptor. That means, when it sits onto the receptor, there is a system called G protein which will be activated. Now, what is this G protein? The G protein is like this, that if you have, this is your membrane, you have this, suppose the G protein coupled receptor. So, there is a receptor site, your acetylcholine sits here. What is G protein? G protein is something, it is a trimer of alpha, beta and gamma, these three subunits. That was that is present as a trimer. So, it is a heterotrimeric trimeric protein, which is also membrane, which is moving into in the membrane, okay, going from here to there. Now, one is called alpha unit, another is called beta unit, another is called gamma unit, and in the alpha unit something is attached which is called GDP, that means guanosine diphosphate. Okay. So, when this acetylcholine receptor binds onto this muscarinic receptor, acetylcholine binds to the muscarinic receptor, there is a binding pocket here where this G protein, why this is called G protein? Because it is attached to GDP. Which one is attached? It is the alpha subunit attached to GDP. Now, what happens? As soon as this binds acetylcholine, this goes and alpha binds to this other active site pocket, which is intracellular, and after binding, see what happens here. It is, we are showing from here. So, the ligand is bound to the receptor, the alpha subunit is bound to the inner side, interior of this membrane, and this is the G protein. And now, what happens? As soon as it bounds, there is a reaction where GDP is now replaced by GTP. So, earlier it was GDP, now GDP comes off, falls off as it binds, and then GTP goes and binds to the alpha subunit. But that has got some that actually dissociates the whole thing from this membrane, dissociates the alpha, falls off along with GTP, and the protein, now this protein which was earlier consisting of alpha, beta, gamma, now the beta gamma unit stays intact, but the alpha is now attached to GTP, and then there is a reaction when the alpha subunit it does a reaction in which you have, let me see, no, it is not there, it is, no, it is not here. Let just, I will write the reaction, that alpha GTP then activates another protein which is called adenylyl cyclase. Adenylyl cyclase. Adenylyl cyclase is basically if you take ATP, that ATP is converted into CAMP. I will write what is that CAMP. So basically again I repeat, because this is a little bit complicated. So first you have this alpha, beta, gamma, the G protein floating into in the membrane, traversing in the membrane, and you have this muscarinic receptor. Okay. Acetylcholine comes and binds to its receptor pocket, and what it does, it opens up another pocket in the same membrane protein, but when it opens up, this alpha subunit binds to this open port. Okay. And as soon as it binds, the GDP falls off and it is replaced by GTP, and then as a result the whole thing dissociates from here, and then and also this acetylcholine also goes off. So, that closes this pocket, and so this whole thing dissociates off, but they dissociate off in such a way that the beta gamma unit remains intact and the alpha is separate, it is attached to the GTP. Now, this goes and activates an enzyme called adenylyl cyclase. Now, what is the reaction that it catalyzes? It catalyzes this is adenine, it catalyzes this reaction phosphate, phosphate, phosphate. So, it catalyzes. So, you have so, this is what is cyclic AMP. CAMP means cyclic AMP. Now, this is called a second messenger, second messenger, and it carries out the signal, and then like activation of adenylyl cyclase. So, you get CAMP, and then that ultimately creates a lot of biochemical reactions which is responsible for cell growth and motility. There are other parts are here, but we are only talking about this activation of adenylyl cyclase. So, again basically I repeat that first it is the membrane protein, the acetylcholine sits, activates it, the G protein binds, the alpha subunit binds, the GTP falls off, and then it is bound to GTP, then the whole thing dissociates, and then the alpha subunit remain alone along with the attached to GTP, and that goes and activates adenylyl cyclase. So, that you get this reaction, you get CAMP. CAMP is called a second messenger, because this is not the primary one. The primary one is the acetylcholine. Because of the acetylcholine, you are releasing CAMP, and this CAMP now goes and takes out the signal and controls the or creates a cascade of processes like activation of pKa, and which ultimately results in cell growth and motility. That means the survival of the cell. Okay. So, this is the two receptors, muscarinic receptors is a G protein, is a GPCR, and the other is nicotinic acetylcholine receptors, and that is ion channel, ligand gated ion channels. Now, the next question is how to control or who controls the concentration of acetylcholine, because that is very important, because that will decide how long this signal will percolate. Okay. So, I told you about one enzyme that is called acetylcholine esterase, which hydrolyzes the acetylcholine and disturbs the equilibrium to the left side. That means the resting state of the membrane protein. Okay. So, this enzyme is very important, acetylcholine esterase. Now, suppose in some cases that the acetylcholine is hydrolyzed more rapidly than is expected. That means, you have more concentration of acetylcholine esterase, and that is hydrolyzing the acetylcholine much before the expected contact time of the acetylcholine with its receptor, then what will happen? You have a problem with the signal processing, the signal transduction, the amount of signal that will be generated. So, there you talk about the disease conditions. That means, the acetylcholine has to be properly balanced, the concentration, so that the optimum level of signal transduction takes place. If the acetylcholine esterase is overactive, then what will happen? You will have less concentration of acetylcholine, because you are hydrolyzing that, and that means you have less amount of signal that will be generated out of this binding to the receptor. So, this is a very important enzyme, and as a consequence, if you stop acetyl, if you there are two ways of doing this. Other is, if the acetylcholine esterase, if you suppose inhibit the acetylcholine esterase completely, shut off the acetylcholine esterase activity, then what will happen? That will also a catastrophic, because you will have the acetylcholine will be bound to the receptor for a long for a greater length of time, and that also causes lot of neuronal diseases. Okay. So, we will talk about that, but before that a quick look at the mechanism of hydrolysis, the mechanism of acetylcholine esterase. Now, we know that it is a serine esterase, serine based esterase. We have read in the first part of this course that how serine proteases work. It is very similar. Serine esterases work by, you have this histidine base, and then histidine activates the serine, and the serine goes and attacks the carbonyl, and resulting in hydrolysis of the forming, first of all forming the tetrahedral intermediate, and then later on hydrolyzed by water. Okay. And that is the mechanism of acetylcholine. It is very similar to what we have read earlier. Now, see, the diseases that are associated with this disbalance of the acetylcholine, one is called myasthenia gravis. It is an autoimmune disease which results from antibodies that block or destroy nicotinic acetylcholine receptors. So, if you destroy the nicotinic acetylcholine receptors at the junction between the nerve and the muscle, okay, the muscle cells at the junction where you have this nicotinic acetylcholine receptor. So, that basically controls the muscle contraction processes, contraction and expansion processes. Then what will happen, that if you, it is an autoimmune disease which destroys the receptors. So, if you destroy the receptors, that means you are no longer having the ligand gated ion channels, and so your muscle contraction will not take place. Okay. This prevents nerve impulses from triggering muscle contractions. Okay. So, that results in this type of eyelids. Eyelids is covering most of the eye, which is exposed to outside. That means, through which we see, that portion is tiny in size. So, most of the time it is almost in closed condition. Okay. So, that is called myasthenia gravis. That means, there is a problem of opening it. That means, because to open and close, you have to have a muscular contraction and expansion. Okay. So, that is controlled by this nicotinic receptors, means nicotinic receptors which are part of the cholinergic receptors. So, this is one disease which is acetylcholine, which is dependent on this acetylcholine receptors. Now, myasthenia gravis is generally treated with medications known as acetylcholine esterase inhibitors. So, now you can take, there are drugs which can treat this, and that is these are inhibitors of acetylcholine esterase, because whatever small amount of receptor sites are still available, if 100% are destroyed, then you get complete shut of the I, but if some receptors are still there and some receptors are destroyed by this autoimmune response. So, what you want that acetylcholine should be there as much as possible. So, that most of the receptors can interact with the acetylcholine. So, that means you have to inhibit the acetylcholine esterase, okay, so that the acetylcholine concentration is more there. Okay. Now, acetylcholine esterases as anticholine esterases, okay. Basically, they are inhibitors of acetylcholine esterase. The enzyme that hydrolyzes acetylcholine, if acetylcholine is not hydrolyzed, is not destroyed, it can return to active to reactivate the cholinergic receptor and increase cholinergic effects. Okay. Therefore, an acetylcholine esterase inhibitor will have some same biological effect as a cholinergic agonist. Means if you like muscarine, suppose you are not producing acetylcholine, okay, in the body. So, what you can do, you can take muscarine, and now muscarine, because it binds to that muscarine is G protein, muscarine is G protein coupled receptors, and if there is a problem with that, you can take muscarine and that will activate your the receptor side GPCR. And if you have problem with this this contraction, then what you can do, you can take nicotine also, because nicotine will also be a will act as a receptor and produce the same kind of signals. So, that means they are agonist. What are agonists? I will tell again. Agonists are basically go to the receptor and then produces the same type of effect like the natural ligand. In this case, the natural ligand is acetylcholine. Okay. The nerve gases diisopropyl fluorophosphate and sarin, these are very dangerous compounds, were discovered and perfected long before their mode of action was known. Basically, what happened, that you produce something and then you see that it has got some biological effect, and you do not know why it is producing the biological effect. So, basically these were first made and then showed that they have very disastrous effect on living systems, but the mechanism was not known, but later on the mechanism was discovered, and basically what have been found, that both agents inhibit acetylcholine esterase irreversibly, phosphorylating the serine residue at the active site. So, if you look at these structures, diisopropyl fluorophosphate and sarin. Sarin is again a fluorophosphonate, because it is a carbon now, fluorophosphonate with the isopropyl group here. So, what happens, that you have this, basically it is serine and then you have a base which is imidazole. The mechanism is that the base abstracts the hydrogen, the serine attacks the carbonyl group of the ester, and subsequently the ester gets hydrolyzed. That is the mechanism. But instead of acetylcholine, what happens, now this fluorophosphate goes. So, now this serine is here. So, that attacks the fluorophosphate, and let us see the, yes, that attacks the fluorophosphate. Here it is shown, serine attacking the fluorophosphate. Of course, there is a base here, that is the imidazole, that assists this nucleophilic substitution, but now the fluorine will leave, because it is a good leaving group. So, F minus will leave and you get a stable phosphate, okay, stable phosphate, that will be difficult to hydrolyze. So, basically you are now, this is irreversible inhibition of acetylcholine esterase. Okay, so that is why they are showing their action. Actually, what happens now, you will get what will happen if it is if you are exposed to this, sarin is a gas, that is even problematic, because gas can be circulated from one place to another very quickly. Okay. So, if you are exposed to sarin, you are going to die within very quickly. Because of this, they are very potent irreversible inhibitor of acetylcholine esterase. Okay. So, that means huge amount of acetylcholine is generated in the synaptic cleft, and that is not good, because that sends constant signal which ultimately paralyze the living system basically. Now, it is always not bad. Serine is very bad if it is used for the destruction of the human being. Okay. But you can also have acetylcholine esterase inhibitors to kill the insect or pests by using again the insects also use acetylcholine esterase. They also have acetylcholine esterase in their body, acetylcholine is there. So, you can actually develop compounds which are extremely important agrochemicals. Okay. They are the ones which are called insecticides, because they kill the insects. Okay. What is the mechanism of their action? They are, see, these are well known compounds, malathion, parathion, and then chlorpyrifos. These are basically all insecticide. So, what will happen here? The similar, very similar, like this nitro derivative, paranitrophenol derivative. So, your serine is going to attack here, and this goes out, and then it stays there, which as the phosphate, thiophosphate in this case. Okay. Thiophosphate. So, the mechanism is very similar, that they are not hydrolyzed quickly, and in the process the pest is killed, the insect, it gets destroyed. Okay. I think the mechanism. Now, the question is, suppose somebody is exposed to this sarin gas, is there any antidote for it? Means what will be the antidote? Because sarin or this diisopropyl fluorophosphate, what happens? The serine is now hooked up as the phosphate or the phosphonate. Okay, the fluorine leaves. Now, the question is, it is an irreversible inhibition, but can this be again reversed back to the acetylcholine and acetylcholine esterase can be freed from that irreversible complex? Now, yes, it is possible, but before that I forgot to mention one thing, that acetylcholine, the structure is this OCO CH3. Now, what has been found, because this is a substrate, so enzyme is having two sides. In one side there must be a negative charge, so that this NMe3 can form a weak bond, and in the other side you have this active serine. So, the reactive serine hydrolyzes it, and this actually offers the binding pocket for the NMe3 plus. Okay. So, if you want to regenerate again the irreversible complex here, that you can regenerate the enzyme from the phosphate, from the serine, which is now blocked as a phosphate. How? That you make a compound like this, a pyridinium based compound, where there is a positive charge here that acts like the trimethyl ammonium salt of the of acetylcholine, and this is the nucleophile. Now, you do not have any ester group here or a phosphate group here, instead you have a nucleophile. Now, the whole thing actually the idea came from the fact that if you are exposed to sarin, so you get this type of phosphate. If you are exposed to sarin, then it was found that this enzyme can be freed by using hydroxylamine. So, if hydroxylamine can generate, because this can attack and then free the serine moiety. So, but hydroxylamine will be very non-specific. It is a very small compound. It's also, it may be quite toxic also, because it is indiscriminately attack at different places. So, which is not good. So, what you want is a very specific one. Hydroxylamine does not possess the part which goes into the which participates in the binding process, that is the positively charged nitrogen. So, what you do, you take an oxime instead of hydroxylamine, you take an oxime based on a pyridinium moiety. Okay. So, you have the pyridinium moiety which forms the salt bridge here between the carboxylate, which is the anion, and then the hydroxylamine is rightly placed into the so that it can attack the phosphate and regenerate the serine. So, if somebody is exposed to serine or any type of nerve gas, then the antidote is this one, pralidoxime, and the mechanism of action is regeneration via nucleophilic attack. But you cannot use any nucleophile like hydroxylamine, which also generates. You want to have the binding partner also. So, in the form of this methyl pyridinium salt. Okay. So, that this is what is which is used for myasthenia gravis, that I told. Physostigmine is the one. Here this is protonated in the pH that we have in the biological medium. So, that acts as the creates the salt bridge, and this is this is an inhibitor. So, here it is the serine, and then serine attacks and releases this part. Okay. And the serine is blocked as this carbamate. So, that is how the this physostigmine, this is a natural product that works against acetylcholine. Okay. So, now what we have, that acetylcholine can be, this is the mechanism of this physostigmine, just here it is written as AR, but the mechanism is same. I said that ultimately you will get a carbamate ultimately. Okay. So, that is stable. So, the acetylcholine you can use it for just to summarize now, acetylcholine is a neurotransmitter. It has got two types of receptors, nicotinic receptors and muscarinic receptors. One is your GPCR, that is muscarinic, another is your ligand gated ion channel, and you can there is an enzyme which maintains the optimum concentration of the acetylcholine, and the enzyme is called acetylcholine esterase. And if you can inhibit this, you can have different kinds of drugs, and depending on the different type of use. If you are using this eye problem, myasthenia gravis, then you use it in human, and that actually can solve some of the problems associated with myasthenia gravis. You can use it also to kill humans, very reactive compounds which are called serine or fluorophosphate. And if you want to utilize it for the beneficiary, for benefit of mankind, then what you do, you actually kill the insects by using it on the agro field, and where the insects are killed by inhibition of your acetylcholine esterase in the insect. And the last point is that how can you recover if somebody is exposed to serine? Then what I said, that you can use oxime based pyridinium salt, which can recover, which can release the serine from the which is blocked as the phosphate, and then thereby the person may be saved. Okay. So, that is all for acetylcholine. Thank you.